All right, good afternoon, everyone. We wanted to do this little webinar today. I've got Dr. Brosnan with me from University of Tennessee. Dr. Brosnan, how you doing today?
Good, Bill. How are you?
Good, good. So really excited to bring this presentation to you today. We're going to spend about 10 or 15 minutes talking about a little innovative adventure in research AQUA-AID solutions, and Dr. Brosnan went on. You know, a couple things I talk about in my technology talks is one, innovation is not waiting for the person next door to do it. And I think that's really important to move the industry forward. In fact, Jim, you and I had a conversation about that earlier this week. And so we embarked on this research with you all, and we really think it was innovative. Involved worm-powered turf, and not just the results we got from worm-powered turf, but it was the process by which we did the research was pretty innovative. So what I'd like to do is just, Jim, hand it off to you, and why don't we talk about how we did the research first, this new innovative method that I don't think we've seen in the turf industry yet. We may be a first in the turf industry to look at root analysis like this.
Yeah, I think that's right, Bill. And let me share my screen really quick here so we can bring up some background literature just to get everybody acquainted. I really appreciate the opportunity to just be involved in this. AQUA-AID came to me, I guess it would have been late 19, with, an idea that they wanted to do a project to look at the effects of worm-powered turf on rooting. And, I was interested in doing that, and, luckily had formed a relationship with some colleagues at a group called Phenotype Screening that were experts in plant root analysis. They had never worked with turf, but they've worked with corn and soybeans and a number of different plants, and all they do at that lab is study rooting and rooting of plants under different conditions. And I thought that this could be really neat because I've seen and done myself plenty of rooting studies that just haven't given the results that you really expected. You didn't have enough power or enough samples to confirm a hypothesis to the level that you would want as a researcher. And we have limitations in a field setting with variability, right? Where you think about a putting green and there's a lot of moving parts there that, that soil is not going to be normally distributed. Nutrients throughout that profile aren't going to be normally distributed, nor is water or particle size distribution or organic matter content. You name it. There's a lot of things that can vary, which in a research context makes it really hard to detect a true treatment effect. So what we did was we took this inside into a lab setting at Phenotype and really attempted to remove all that background variability to give us a clear look at what worm power could do to turfgrass.
So let's talk about the process by which we did that in this Phenotype Screening Corporation. So you had mentioned all they do is study roots. That's pretty much all they do. They analyze the roots several different ways, but you had mentioned by eliminating the variables. Let's talk about how did we eliminate the variables in this research with phenotype screening, and we can move to the bottom of the screen there and talk about the artificial root zone versus a soil profile. And I know you tried to do a little bit of this with a soil profile. We got just too much attenuation in the X-ray, even with as much washing that we did. So let's talk about the process and the company and how we obtain these X-ray images.
Yeah, no, that's a good question, Bill. I mean, the process is fascinating, in my opinion. What Phenotype has developed is they essentially grow plants, and in our case, turfgrass. We picked annua bluegrass as the species to work with here. In an artificial root zone, and that artificial root zone is made up of polystyrene beads. Those beads are about the size of a medium to a very coarse sand particle, and it forms a true profile, as you can see in the photos below. The easiest way to really think about this, I think everybody has seen the old coolers that were styrofoam, if you will. If you were to break one of those apart, or sometimes you see it in packaging material, if you break that apart, those individual little beads is what this profile is made of. And what makes that really cool in an agronomic context is it provides all of the architectural support and stability that a USGA spec root zone or any sort of soil profile would provide, but it doesn't have any of the nutrients, right? There's no nutrients in the system. There's no water in the system. It's just a material that gives that architectural stability. And what makes it unique is that when the phenotype team can run an X-ray through it, as we see in the photos, the polystyrene does not show up on X-ray. So it gives us the ability to look at rooting through the profile with essentially removing the profile itself. So we capture only the plant material, and that allows us to remove all that variability and truly look at the effective and applied treatment on our species of interest.
And you and I have spent a lot of time washing plugs. I mean, we've done quite a bit of worm power turf research over the years, and the current method prior to this was, set up your plots. You have your controlled and your worm power, and to your point with variables, I mean, you could have variation all over that plot, especially when you're talking about fertility, soil biology, soil structure. We removed all of that. To gather the data here and the images you're seeing down there, you didn't have to pull a plug, right? So let's talk about the project itself in terms of, you mentioned it was a POA. It was in an artificial root zone, but we did need to supply some nutrients. So there's a control, and then there's just worm power turf, but both of those replications had nutrients applied to the plants, so that way we had a healthy POA plant, correct?
Yeah, that's right. So everything in the experiment, and we had substantial replication in the study, everything got a 75% Hoagland solution applied as fertigation. Hoagland solution, for those that might not be familiar with that, it's essentially a nutrient solution that delivers the nutrients required for plant growth, both macro and micronutrients, into the system, and we fertigated that over the top. And then in our worm power treated profiles, we would inject worm power into that nutrient solution as well and deliver that. I believe our rate was an 8.5% solution via fertigation. And, it's interesting going back to the harvesting cores thing. When I originally approached Phenotype, the concept to save time was, well, we could bring cores from field plots into the system. And there was some doubt about whether or not we could get them clean enough that they would show up in the X-ray. Because if we had mineral matter from the soil, then that would kind of not give us a clear X-ray picture. And we wouldn't be able to see what we wanted to see. And I harvested plugs myself. I didn't have my technicians do it. I didn't have graduate students. I harvested the cores myself. I spent over an hour per cup cutter washing these cores. I walked into this lab with my chest puffed out thinking I had cleaned these as clean as one could possibly get them, and we would be able to use them in this X-ray system and go direct from a putting green into the lab. And I was awakened very quickly that my 1 hour per core washing was not clean enough to eliminate the mineral material such that we could run X-rays through these. So we made the decision that we would germinate annual bluegrass seedlings, transfer those germinated annual bluegrass seedlings into these profiles, and then grow them in these profiles until they were mature. And then at that point, begin our experiment with worm power in nutrient solution in combination.
So we've got some great images down there at the bottom, and images are great. And for years we've had, people on social media. I mean, it was really how worm power launched. I mean, people were taking photos of their control areas and areas they had applied worm power turf, or they said, this year the only thing I did was add worm power turf. I didn't change my program at all and look at my roots. And that was great, and that's some anecdotal research. And here we have images, and that's great. But one of the other things that this phenotype screening was able to do is I would equate it more to a CT scan, where they can actually take measurements of the root mass, root area within these columns. And I think that is really the innovation, besides the images here, but really the innovation behind this research. So let's move over to the graph in the top right there, Jim, and I'm just going to let you run with it because the numbers were unbelievable that we saw, and the process too, that we saw in terms of results with these plants treated with worm power turf.
Yeah, no, I think that's right, Bill. And, the data and the graph kind of mirror the images pretty nicely. When you look at the images, when we think about rooting studies, everybody wants to focus on length, and there are some length differences here. To me, what I see in those photos is just kind of a general abundance and mass of roots, and that continuing as you get deeper into the profile. And the data in the graph at the top right really show that. So what we did was we have these scans, and then we can use software technology to essentially put some real numbers on length of roots, area of roots, diameter of roots, and the graph presents cross-sectional area per profile depth. So the abundance of roots, or the area of roots for every inch of the profile. The top of the graph is 1 inch depth, and then we go to a 2-inch depth all the way down to a 12-inch depth, the bottom of what a USGA root zone would be. And the green bars are our control plants, and the blue bars are our worm power treated plants. And we had 6 replications in this study, and then 4 subsamples per replication. So we have a lot of experimental units here. You can see standard error bars on each one of those to statistically separate worm power treated from our control. And you can see as you go down through the profile an increase in our overall root area almost at every inch down in the profile as you go. So we had 1.5x more root area at the 1-inch depth with worm power treated with worm power treatments. At the 2-inch depth, we have 2.1x more root area. 3-inch depth, we have 3.1x. 4-inch depth, we have 8.3x, and then down at the 5-inch depth, we have 17.6x. It's really it was surprising to me. I mean, I've never worked with worm power before. This is kind of my first look at the product. I've certainly seen the social media posts that you referenced earlier, but never having run a study with it, I was pretty surprised with these results.
So when we look at this graph up here, you can see this is after 88 days of exposure. So going back to what Jim had mentioned, we had grown, Phenotype Screening Corporation and Jim had grown these plants to maturity, and then the worm power turf was introduced. So the control was the complete nutrient solution, and then the worm power was a complete nutrient solution, and the worm power turf. It was essentially fertigated over top of the plants and then down into the profile for the roots to absorb. But this is after 88 days, and we talk about worm power turf needing to be on a program. This wasn't a one and done. A couple days later, we took these images. This was a program approach, like we talk about with worm power turf. So what you're seeing here is data after 88 days, which is what we tell turf managers that want to go on a worm power turf program to do. It's important to be on a program. This is a biological. These are microbes, and you can see when you introduce worm power turf the effects you get in these root zones. Jim, there was some. Oh, go ahead, Jim. I'm sorry.
I was going to say, Bill, and you bring up a really good point with the idea that it's biological. And again, I was a worm power novice when this all started, and I kind of learned that as the project went along. And Dan McDonald, who's the lead at Phenotype, lead scientist there, we had some reservations about this system with these polystyrene beads is relatively inert, and we're supplying our nutrients through fertigation. Would there be enough there for activation? And there clearly was, based on not only what we see in our X-ray images, but in our data. And, you'd have to do more work in a different environment or to prove this theory. But I would think, looking at this, that if you put this into a living root zone with more food for those microbes to utilize, the response could even be greater.
Yeah, yeah. So there was some other data we collected, particularly the two charts on the bottom there, Jim. The mean root area after 88 days. If you want to talk about that, and then also the dry weight. The dry weight is something we again have always tried to do. Some folks anecdotally, and then in labs, but you always have that the variable of contamination. Have you gotten all those particles off? As you said, you thought you had a clean sample and then put it in the x-ray and there was attenuation. So let's talk about those two as well and the results we saw from that.
Yeah, I mean, I think that the root area there is just a compilation of what we've talked about so far as we go through the profile depth and then with the x-ray images. I mean, the root dry weight at the end of the day, that's really important, right? Because what we see data-wise and is generated from software, and software has its limitations and whatnot. The dry weight's a real quantitative measurement. That's not a calculation by a computer. That's taking the roots off those individual poa plants, getting them to a constant dry weight, and then measuring them. And it shows the same thing that we see with our software-driven analytical measurements. We see with a dry weight measurement, and again, in this controlled system where we've removed all the variability that can be out there and looked clearly at what is the effect of worm power on annual bluegrass, we see a real clear response with almost a 2x increase in root dry weight after 88 days of exposure.
Yeah. Well, Jim, this was really, I think, a leap of faith for all of us. Again, the process by which this data was obtained, it may be being done in laboratories and for what I would call, from a technology standpoint, beta testing things, but really never been published. And when you and I talked initially, we knew this was sort of a leap of faith in terms of the technology. We at AQUA-AID Solutions know that worm power turf works. We wanted something definitive to say, yes, we can increase your root area. And all the other data that you just presented, we knew we would come out on the positive end of this, but it was the technology behind this that we were really taking a leap of faith, and it was exciting to do this. Looking forward to more, but I don't know if there's something you wanna close on with that.
Yeah, I mean, I just think it's another example. I've known you, Bill, a long time, and you've always been a technology guy, and really, one who prides themselves on innovation and bringing solutions to the industry. And I know AQUA-AID feels the same way as a company. They're always trying to be creative about bringing solutions to superintendents, from what you all do with wetting agents and products like worm power to helping put on, digital annual bluegrass control field days, right? That's just another way of giving a superintendent a solution. And to me, this is a really good example of it. Like you, I had some heartburn. The Phenotype team had never worked with turfgrass, and we had some doubt about whether turfgrass would even set up for the system, and ran several pilot studies to make sure that we had enough confidence to go forward with a big project with worm power like this. And I'm glad we did. The results turned out really, really well, and it's really cool technology.
Jim, appreciate you joining us this afternoon, and look forward to talking to you again and your time on this research. Thank you.
Thanks for the opportunity, Bill.